Lesson 3.2 Anatomy of a Protocol. A real protocol can be anywhere from 50 to 200 pages long. What's inside all of that? In this lesson, we'll take a guided tour through the 12 core sections that appear in virtually every clinical trial protocol, giving you a practical map of the document that will become very familiar over your career. 5 minutes. Let's open it up. Here are the 12 core sections you'll find in virtually every clinical trial protocol. Sections 01 and 02 establish the identity and rationale of the trial, the title page, protocol ID, and the background and scientific justification. Section 3 is objectives and endpoints, the questions the trial is designed to answer, and exactly how the answers will be measured. Sections 04 and 05 define the structure and participants, the study design, and the eligibility criteria. Sections 6 and 7 are operational, treatments and the schedule of assessments. Sections 8 and 9 are the two most technically demanding, safety monitoring and statistical methods. Section 10 covers data management. Section 11 addresses ethics and regulatory requirements. And Section 12 contains appendices, often including the informed consent form template and investigator references. Different sponsors use different numbering and different titles, but these components, or something very close to them, appear in every protocol. In the next several slides, we'll walk through the most important ones in detail. Let's start with Sections 1 and 2, the administrative foundations. Section 1 is the title page and protocol identification. This seems simple, but it's actually critical. The protocol number is the unique identifier for this study. You'll see it on every document, every case report form, every regulatory submission. The UDRCET number in Europe or the IND number in the US links the protocol to its regulatory tracking record. And critically, every protocol has a version number and a date. Every time the protocol is amended, a new version is issued. During an audit, you must be able to confirm that every site was working from the correct version at every point in time. An outdated protocol at a site is itself a finding. Section 02 is the background and rationale. This is where the sponsor makes the scientific case for running the trial. Why does this disease need a new treatment? What did the preclinical studies show? What happened in earlier phases? What is the mechanism of action of the investigational product? And most importantly, what is the benefit-risk assessment that justifies exposing human volunteers to this compound? The background section is also the historical record. If you join a trial 2 years after it started, Section 2 is where you go to understand why. Section 3 is one of the most important in the entire protocol: objectives and endpoints. First, the objectives. Every protocol has one primary objective, the single most important question the trial is designed to answer. Everything else in the trial is optimized around answering that one question. There is only one primary objective. If there were two, neither would be answered with full confidence. Secondary objectives are additional questions of interest. The trial generates useful data about them, but it's not formally powered to answer them definitively. And exploratory objectives are hypothesis generating, things like biomarker analyses or subgroup effects that might inform the design of a future trial. Now, what's the difference between an objective and an endpoint? The objective is the question. The endpoint is the measurement used to answer it. For example, the primary objective might be to evaluate the effect of drug X on overall survival in patients with non-small cell lung cancer. The primary endpoint is overall survival, defined as time from randomization to death from any cause. It's specific, it's precisely defined, it's pre-specified before a single patient is enrolled. This pre-specification is everything. If the primary endpoint is vague or ambiguous, if it can be measured in multiple ways, regulators will reject the analysis. And if you change the primary endpoint after unblinding, that's scientific misconduct. Sections 4 and 5 define the architecture of the trial and who can participate. Section 4, Study Design, covers the fundamental structural choices. Is this a randomized controlled trial or a single-arm study? What's the randomization ratio? 1-to-1? 2-to-1? What are the stratification factors? Is it blinded? And if so, who is blinded to what? What is the control arm? How long is the treatment period and the follow-up? These choices directly determine the quality of evidence the trial can produce. A double-blind, randomized, controlled trial produces the highest quality evidence. An open-label single-arm study produces weaker evidence, which is sometimes the only practical option, such as in rare cancers where a placebo arm would be unethical. Section 5 defines the study population through inclusion and exclusion criteria. The inclusion criteria are the characteristics a patient must have to be eligible, things like age range, confirmed diagnosis, adequate organ function, appropriate performance status. The exclusion criteria are conditions or characteristics that disqualify a patient: prior treatment with the same drug class, active infection, pregnancy, prohibited medications. Together, these criteria define the exact population in which the drug will be studied. Get them wrong, too broad, and you enroll patients who can't tolerate the drug; too narrow, and the results won't generalize to the real-world population. This is one of the most scrutinized sections at regulatory review. Sections 6 and 7 are where the trial becomes operational. Section 6, Treatments, covers everything about what patients will actually receive. The investigational product, its formulation, strength, manufacturer, and storage conditions, the dosing instructions, dose, route of administration, frequency, and duration, all stated precisely. Dose modification rules, what to do if a patient experiences toxicity: hold the dose, reduce it, or discontinue. Concomitant medications, what's allowed while on study and what's prohibited and why. And compliance monitoring, how adherence will be tracked. Section 7 contains what's called the Schedule of Assessments, often abbreviated as the SOA. This is a table that lists every single procedure, test, and visit in the trial, alongside the exact time points at which each must occur. Every clinic visit, every blood draw, every imaging scan, every patient questionnaire, every ECG. The SOA is the document that site coordinators live by day to day. It tells them precisely what to do and when. If an assessment is performed outside the defined time window, that's a protocol deviation. If it's missed entirely, that may be a major deviation. Of all the sections in the protocol, the SOA is often the one that changes most frequently across amendments, because as a trial evolves, the operational requirements frequently need updating. Sections 8 and 9 are the most technically demanding parts of any protocol. Section 8, Safety Monitoring, starts with definitions. What exactly constitutes an adverse event? What makes an event serious and therefore an SAE? The protocol must define these precisely because they determine reporting obligations. A serious adverse event must be reported to the sponsor within 24 hours of the site becoming aware of it. A SUSAR, a suspected unexpected serious adverse reaction, must be reported to the regulator within 7 days if fatal or life-threatening, and 15 days otherwise. Section 08 also defines causality. Was the event related to the study drug? And expectedness. Was it listed in the investigator's brochure? Unexpected and related events trigger the most urgent reporting pathways. And the section defines stopping rules, pre-specified criteria that trigger pausing a patient's treatment or halting the entire trial. Many trials also have an Independent Data Safety Monitoring Board, or DSMB, that reviews unblinded safety data at predefined intervals. Section 09, Statistical Methods, is where the biostatistician's work lives. Sample size and power, how many patients are needed, and what assumptions justify that number. The primary analysis method, the specific statistical test that will be used. The analysis populations, who is included in which analysis, intention to treat, per protocol, or safety populations. How missing data will be handled, and the plan for any pre-specified interim analyses. Regulators scrutinize Section 9 intensely. Any ambiguity is a red flag.